until it's really a pleasure to be here. How's the lights are getting off. So I'll start by congratulating Yael on the new spirit, continuing the very nice activities in the Nano Center. Zostabish is also here, and I think that we see a continuation of changing and very good spirit and activity in this center. As Yael said, I've been a member of the center for many years, organizer of one of the previous meetings, speaker in some of the meetings, so I have a complicated task in try to give for the newcomers some idea about the things that we do, and while providing the veterans some new stuff. So I started, as was asked, with some description. Is there a pointer by any chance? As you could see, we are working on different things that ranging from applied chemistry to new materials to general nanotechnology, chemical biology, and the common denominator between... Oops, למה זה לא עובד? לא פה ולא פה. זה לא עובד לא פה ולא פה. לא חושב. לא ידעתי שאני הייתי כאן. כן, כן. זהו, נעשה. Is the study of molecular recognition and self-assembly at the nanoscale. We are interested in small building blocks, 500 Daltons and smaller. We study organic entities, including biomolecules, hybrid molecules, such as peptide nucleic acid. You could see the poster of O. Studying also synthetic novel chemical entities. For the physicists, these are entities that were not there in this world before we designed them and synthesized them. We utilize a diverse set of methods, including chemical synthesis, chromatography, microscopy, both optical and electron microscopy, spectroscopy, X-ray and electron diffraction, molecular biology, animal models of ranging from chemical methods to physical methods to biological methods. And the subject, as you will see today, are including physiological and pathological studies related to molecular self-assembly, viral self-assembly. I won't speak about it today, but this is another interesting topic in which we see a very elaborated process of self-assembly that could be studied at the nanoscale as well as manipulated, namely development of antiviral drugs, protein thermodynamics, drug development, new materials, and more. As for the methods we use, as I said, it's diverse, including a lot of peptide work, including a peptide array, synthesizing of tens and sometimes hundreds of peptides on solid support, a synthetic peptide and spectroscopy, also biological a presentation of different structure, electron microscopy, both fly and mice model, X-ray and electron diffraction, and NMR. So I'll start with our main interest in the last 12 or 13 years already, which was trying to understand a self-assembly process at the nanoscale that is related to very significant human disorders. And this is the formation of the amyloid nanofibers. These are fibrillar structures of 7 to 10 nanometer in diameter, that their formation is associated with major human disorder, including Alzheimer's disease, Parkinson's disease, type 2 diabetes, and many more. You don't have to be a biologist or or a medical person from the medical field to know about those diseases. And the thing that is observed in all these diseases is actually the formation of these fibrillar structures by the process of self-assembly of protein molecules. These are well-ordered structures. You can see it from the diffraction pattern with the typical 4.6 to 4.8 angstrom on the meridian. For the biologists, this is containing predominantly beta sheet structures, and for the rest, these are planar structures that could be stacked one on top of the other to form all the structures. And an interesting point for us was the fact that fibers from different origin, let's say from the brain of Alzheimer's disease patient and the pancreas of type 2 diabetes patient, show remarkable biophysical and autostructural properties, those that I mentioned here as well as others, in spite of the fact that there is no simple homology in the level of amino acid, of the building blocks, of the molecular building blocks between different amyloid-forming proteins and polypeptides. The story is even more complicated. I won't get into the details since it's a diverse audience, but still I have to mention, to give, especially to the student, the flavor of about the science in action. I mentioned those semi-crystalline, well-ordered structures with the typical diffraction pattern which along the long axis of the fiber. Actually, we and others, by using various biophysical tools, realize that some structures that either on pathway or off pathway to form these structures are involved with a pathological pathology of these diseases. One thing that was mentioned in the slide, and I want to stress, is the fact that we see colocalization of the formation of these nanostructures with damage to tissues. Let's say in Alzheimer's disease, areas in the brain that are involved in memory impairment. In the case of Parkinson's disease, areas in the brain that are involved with motor activity. In the case of type 2 diabetes, areas in the brain that are involved with insulin production. And the thing that was observed, just to give you the feeling, by using some biophysical response, we could see that soluble structures, and not these fibrillar structures that sediment upon centrifugation, could show some activity that is related to tissue damage. And this is in two different concentrations, and in a lower concentration, as nucleation-dependent self-assembly process that is concentration dependent, we could see the formation and disappearance of transient membrane active structures. And this gets even more complicated. This is not from our group, but another group in Minnesota later on demonstrated, in the case of Alzheimer's disease, the formation of specific structures, prefibrillar assembly before the formation of the nanofibers, other nanoassemblies that are involved in memory impairment in Alzheimer's disease model mice. And the reason that I mention it is because I will show you that we can completely inhibit the formation of these dodecameric structures, structures made of 12 building blocks that are being formed in this case. So the key question for us, and this is the study into nanoscale project, is try to understand the molecular basis for the recognition, which follow the self-assembly process that lead to the formation of the structure, with special interest in the very early stage that lead to the soluble nanostructures. And I won't get into the experimental details, but just to give you the highlights and the rationale. So, in the past, the previous century, it was known that these structures are formed by proteins and polypeptides that are made of 30 or 40 amino acids or even longer. Our approach is a reductionist one. We try to find the smallest fragment that can form amyloid fibers, and we are quite proud to set time after time some even world records for the smallest fragment that can form these structures. And by finding small structures and using synthetic chemistry, we could change in turn the different residues and pinpoint residues that play a role in the molecular recognition and self-assembly process. Based on these insights, we identify new fragments, even shorter, and develop new way to control this process, namely a drug candidate. I will start with the first example already, more than 10 years ago, in which it was a little bit like a detective work. This is why I go to a rather early study. This is fibers that are being formed by the human calcitonin. This is a hormone that is found in thyroid carcinoma, a cancer of the thyroid tissue. In this case, we knew that the formation of amyloids by the full-length protein is pH dependent, which led us to believe that charged amino acids that can change the charge state in different pHs could play a role. And then we were quite lucky because there was only 2 charged amino acids. And to make a long story short, we made shorter and shorter version, and we get the first demonstration ever that the pentapeptide can form typical amyloid fibers, and here and even a tetrapeptide, peptide form of 4 amino acids, can form well-ordered fibular structures at the nanoscale. We later on studied many proteins. This is just to give you an interesting point. This is a medin. It's a protein that forms amyloid in these nanoscale fibers in virtually all individual older than 60 years. The importance is not fully understood. It probably has to do with the changing of the flexibility of the aorta, via Okim. And here our contribution again was not in the medical point of view, but rather cutting the protein into small fragments and identify the smallest fragment that can form these amyloids. We found a hexapeptide that can form these structures. And this is a list in using the one letter code for each amino acid that can form amyloid fibers. Most of them identified in our group. There's no a clear common denominator between the different short peptides. But one thing that we realized is that our aromatic amino acids are quite abundant in those short fragments. And we came on with the notion that it might be that these aromatic amino acids, even for the physicists, these planar, sometimes hexagonal structures that you remember from high school chemistry. The important thing about them is that it is well known in supramolecular chemistry, in the chemistry that involves non-covalent interaction, that interaction between these planar entities have specific geometries. And this is a little bit like building Lego or structures, but doing it in the nanoscale. And we came up with a model in which the organization, the molecular organization of such aromatic moieties, known as aromatic leather, could play a role in the arrangement of such well-ordered structures at the nanoscale. This was based on the occurrence of aromatic amino acid, on our ability to change this aromatic amino acid to aliphatic one, and still see the formation of these structures. We were in the dark for a few years, but quite luckily, upon the use of electron and X-ray diffraction pattern, a group in Cambridge demonstrated and gave experimental support to the model that we suggested. And this is now became quite central in this field. thing is to identify short peptide fragment that can form this amyloid, and this is the reductionist approach, finding the smallest fragments. The other direction, after you understand which are the smallest fragment and the chemical interactions that play a role in the formation of the structure, is making all kinds of inhibitors. And we have two main directions. One is based on peptidomimetics. The information is less important, but more the general idea. Other is based on small organic molecules. Again, you don't have to be a chemist, but you could see, once again, this hexagonal ring, and on the other end, these structures, and by steric hindrance, you can have the binding on the one hand, which does not allow further growth of the structures, and this is a direction for inhibition. This is two main directions in our lab. Again, I'm doing quite a quantum jump here to our selection of active compounds from a rationally designed library. Namely, we acted like a small pharma, a nanopharma company, to optimize various structures that can form, that can inhibit the formation of these nanostructures. To the chemist and biologist, this is the dodecameric structures that I mentioned in the beginning, that were found that to be correlated with memory impairment with Alzheimer's disease model mice, and we could show a complete inhibition of their formation. Another quantum jump to after the study of safety, oral bioavailability of these what they were novel chemical entity, as I mentioned in the beginning. We study their efficacy in mice. So how you study memory in mice? So it's mice and maze. You treat the mice to find their way in a maze, and you either measure the time that it takes the mice to find their way in the maze, or the length of the path, because if they don't remember, I'm going to a longer path. And we could demonstrate that upon treatment with our compound, we see a significant improvement in the time that it takes the mice to find their way in the maze after 3 and 4 days of training. Which was quite impressive, but became much more impressive when we added the third group, which are normal mice. They lack the mutation that lead to memory impairment in those mice. So we have normal mice in blue. We have mice treated with our compound in red, and mice treated with vehicle, with placebo, in black. And you can see that either in the length of the path or in the time that it took the mice to reach the platform, there's no significant difference between the treated and normal group, but these two groups are significantly different from those treated with the vehicle. So we could not only significantly improve the cognitive abilities of these mice, but we could also restore them to a level that is similar to non-transgenic mice. Well, at least we know how to cure mice from Alzheimer's disease. But this compound, actually, a derivative, is currently entered just last November. Entered a clinical trial, so it's in phase 1 clinical trials. So maybe in the 14th meeting of the Nano Center, I could report treatment in.
Start using it.
And here, just to give you a highlight about other things that we do, just as appetizer. We are working also on alpha-synuclein, which is associated with Parkinson's disease. And using the same concept, we identified short peptide fragments that can inhibit the structures, doing peptidomimetics work, and studying efficacy in animal models. Actually, also this compound is in preclinical development with an Israeli pharma company. This is a second generation compound that is still in preclinical. Again, to those who are interested more in the chemistry and drug development aspects, one thing that we always do is using NMR in order to identify, and this is in collaboration, a very close collaboration with Debbie Shalev, when was at the Hebrew University. So for each compound, we have we know the interaction at the molecular level, and we study the KD for the binding to this structure. And we are moving from micromolar to hundreds of micromolar, nanomolar to tens of nanomolar. Actually, this compound now have a KD of about 50 nanomolar, and in the past it was questioned whether you can manipulate the formation of molecular assemblies by small molecules. And now this is a paradigm change in the field of pharma, in which we could really manipulate the structure by high affinity ligands to nanostructures. So in the last third of the lecture, I want to tell you the old stuff and new stuff about moving from amyloids to new materials of unique mechanical, optical, and piezoelectric properties. So I told you that we gave the first demonstration that very short peptide, the shortest tetrapeptide, can form typical ordered structures. And similar to that, we study most of the amyloid-forming proteins and polypeptides. And in our path to search for the smallest amyloid-forming structural motif, we studied the co-recognition motif of Alzheimer's disease. I mentioned this peptide already. Starting point was knowing that a heptapeptide, a 7-amino acid fragment, can form this amyloid. 2 pentapeptide inhibits the formation. By now, 2/3 into the lecture, you know that this is the two sides of the balance. Inhibitor usually bind the recognition site, but do not allow further growth of the structures. The common denominator, which should have been the recognition modules, was the diphenylalanine. By now you know that we like these aromatic amino acids, the hexagonal planar structures. So we thought that we'd be the first in the world to show that a dipeptide could form amyloid fibers. Again, it's a 10-year-old story. Quite to our surprise, we realized that these probably the simplest peptide motif that can form the ordered structure, form tubular structures, nanotubes. We use these nanotubes for as casting mold, degradable casting mold, for fabrication of nanometric objects. Moving from ionic silver to elementary silver, then peeling off the peptide structure by enzymes. We ended up with silver nanowires of 20 nanometers. 10 years ago, it was very impressive. Nowadays, we can do this fabrication using lithography, reaching this. Although for the very small elements, interconnect and so on and so forth, it's still a challenge. But I think that the nice thing, and people who are working in the field of nanotechnology, bottom-up nanotechnology, do not need to be convinced that the nice thing is really the ability to form a more complex structure. In this case, like the forming of coaxial nanocables. For other applications, this is actually collaboration with Yael. We wanted tubes that could be for nanofluidics. For example, we wanted tubes that will be resistant to proteolytic degradation, and then, again, not too complicated chemistry, moving from D-amino acids to stereoisomers of D-amino acids. From L-amino acid to D-amino acid, we could get tubes that looks just the same, but are completely resistant to proteolytic degradation. Other twist to the story came with a study of various different building blocks. So I told you that we make nanotubes, but those of you working on carbon nanotubes, they have a diameter of few nanometers. These nanotubes have diameter of few tens of nanometers. So we thought that maybe moving from diphenylalanine to diphenylglycine, we could get tubes of smaller diameters. Quite to our surprise, we got spherical structures, which was also interesting, as now we understand nanostructures many times as we know, with carbon structures and also with inorganic structures, come in two flavors, either tubular or spherical ones. Other direction was made, we made hydrogels, structures that contain more than 99% water, but are very rigid in this case. So again, without getting into all the details, I told you about the starting from biology, going to nanostructures, then studying all kinds of different analogs. We ended with hydrogels and moved to application of a slow drug release and tissue engineering, so back to biology. So one thing is to make the nanostructures. Another thing is to make order arrays of these assemblies. And we've developed few methods. Here it's by unidirectional growth. We make these what we call nanoforests, tens of nanotubes or hundreds of nanotubes that are organized in an array. Another direction was based on the use of low-resolution inkjet, printing these structures. Other directions, this is collaboration with Gil Rosenman, were based on physical vapor deposition. Once again, for the chemist, this was quite a conceptual change in which we moved by aromatic biomolecules by heating to the gas phase and then grow them on a cooler substrate. So I told you about the structure, making the structure, the position of the structure. One thing that we realized is that the physical properties of the structures are quite unique. The very first thing was the rigidity, because you see the persistence length of these structures. And then we collaborated with Itai Russo, who was then at the Weizmann Institute, to directly measure the rigidity of these structures using AFM indentation techniques. We applied force on the structures and measured Young's modulus of about 20 gigapascal. Just to give you a reference, this is like already close to metallic structures. The strongest plastics are about 4 gigapascal. Bone and teeth are about 20 gigapascal. Titanium is about 90 to 100 gigapascal. So we are getting really very rigid structures. And even more surprising results was when we studied the nanospheres, in which we could not even deform these assemblies only with diamond-made cantilever. So these have about 200 to 300 gigapascal, like a good steel. Other properties are optical properties. This is for the physicist. Piezoelectric properties and semiconductive properties of the structures. And we have many technological applications. As time is running, I will skip this one. And just to summarize, amyloid nanofibers are naturally occurring self-assembled biological structures at the nanoscale. We demonstrated the role of aromatic interactions. We speculated on these geometrically restricted interaction to provide energetic contribution and order and directionality the self-assembly of the structures. Suggested new approach to understand self-assembly and identify new motifs and indicate a new way to control this process in drug development. Zooming out in our reductionist approach, we demonstrated that even a dipeptide contains all the molecular information needed to mediate molecular recognition and self-assembly. We use this as a degradable casting mold. We can make all kinds of different architectures at the nanoscale, nanospheres, nanofibers, hydrogels, control the assembly of peptide nanostructures. And I skipped the last item, but recently we demonstrated that at very high concentration, even single amino acid can form all the structure. This is, of course, collaboration with many people. I mentioned some of the collaborations. And, of course, the very talented group of students. I won't tell about each contribution, but I was very fortunate to have excellent students over the years, and this is really the real pleasure. And just a last sentence, although we're running out of time. I think that, as you could see, our work is ranging from biology to physical characterization to chemical synthesis, and I should say that many of the ideas came in those meetings of the Nano Center, and really the interaction between people from different disciplines is so important for the development of a field of research like nanotechnology, and I hope that this tradition will continue. I moved very fast, so those who want to understand the chemistry or biology in more depth could look at my website, which we have a list of the original papers. So thank you for your attention.
So the basic thing is how many other proteins, other than the ones, how many other proteins that contains diphenylaniline outside the brain are there? And what are the risks if you try to inhibit such structures or promote such structures? Why don't we see this fibrous?
Why don't we see the structure? So you can say that there's, if there are 20 amino acids, diphenylalanine, you should see in 1 in 400. Yeah, so there are two things. First of all, aromatic amino acids are among the rarest. It's not that you have 5% of each amino acids. Moreover, aromatic amino acids are usually not exposed to the solvent. They are buried in the core of the protein. It's a completely different field that we also are interested in, which is core packing, the way that the proteins are. So a situation in which you have the diphenylalanine motif exposed to the solution, and you need more than one case to form this structure, is rare, and it is prevented by nature.
Brain. Why is it just happening in the brain that you see that they are exposed?
Why do you see it there? So usually you find it with relatively small peptides, such as beta amyloid or alpha-synuclein, which are partially or fully unfolded. And even that, you could see it only at a relatively low efficiency, and it's not a coincidence that amyloid disease, unlike, let's say, cancer. Amyloid diseases are associated with old age, as cancer and other diseases, but you don't see Alzheimer or Parkinson with children, right? While you see, for example, unfortunately, cancer. This is because, from my belief, this is just a physical stochastic event in the chances to form these structures. And lastly, you don't see it only in the brain, but in the brain the effect is so severe that you see so if you have some damage to a tissue in your arteries or any other organ, it's not that critical. But if you have some damage to things that are related to motoric activity, to memory impairment, you see it. I hope I answered at the multi-level question.
So an important current theory in the neurodegeneration field is that the plaques, the fibrils, may not be damaging themselves. That the smaller oligomeric species would be damaging, and maybe the large plaques are even protective by stealing species, stealing monomers, and inhibiting the formation of the small species of the oligomers. How do you feel about that?
So I mentioned it by passing because I wanted to give the entire story. But indeed, as I said, we were, before it was as popular as now, among those who demonstrated and advocated the idea that it's the early soluble oligomers. And I think that the reason that we could get to clinical development was the fact that our optimization, again I moved quite fast, was actually targeting these early oligomers and not the fibers. And I do probably... in the past, there was a notion that it may be that the fibers are even protective. Actually, now, as we understand it, it's probably diseases like Alzheimer's disease are multifactorial, and it is probably that the fibrillar assemblies are consistent with some chronic damage to the brain, while the soluble structures are concerned with acute problems with memory processes like LTP. I don't want to get into details of in a diverse audience, but processes, specific synaptic processes that are involved with the memory itself, and the not so happy note is the fact that probably it has to do with much more common disorders like something that is called MCI, mild cognitive impairment, which is impairment of short-term memory. I think that all of us above the age of 40 have it. A part of Yael. She's not 40 yet. It's just, you don't remember where you put your keys, and it probably has to do with these early oligomers that are found in.
So I thank you for the sake of being on time, which is just right. We'll thank Udi again. Actually, one interesting comment is that just last month it was announced that a new startup company was established named StoreDot, which is a spin-off of the activity of Gil Rosenman, following up on exactly the same structures, and it's actually pretty funny. I never thought about it until now, that it's for memory devices. So you start from Alzheimer, and you end up with memory chips made of these molecules, so it's pretty nutty.